Hello, dear students. Today, I want to talk about microarray technology, which is again an application part of the biotechnology or recombinant DNA technology. This microarray technology has actually revolutionized the field of diagnostic as well as the study of gene expression, genome study, and all those things. So, an array is an orderly arrangement of the samples where matching of known and unknown DNA samples is done based on the base pairing rules. So, here mainly it is nothing but simple hybridization experiment, you can say. But only thing is, here hybridization is not going to be done only for few genes. Here the hybridization is going to be done for a large number of samples at a time. So typically the sample spot is going to be less than 200 micron size diameter spots, and thousands of spots are going to be made on a single glass slide, or even nowadays people are also using Affymetric matrix. So these thousand spots are going to be known as the probes because they are going to be immobilized on the solid support here, and all these probes are going to be labeled with fluorescent molecules, or sometimes even the radio isotopes are also going to be used for labeling, which further going to be studied by using the particular method of particular technology is going to be used to analyze the hybridization. So here mainly the binding happens based on the complementarity, and an experiment with a single chip can provide information of thousands of genes simultaneously. So this is, you can see the cell, we take mRNA and there will be an array chip is going to be there. On the array chip the mRNAs are going to be added onto that, and it will be incubated for some time, so that after incubation they will wash it off all the unbound mRNAs and then subject it for analysis by fluorescence, if it is fluorescent level. If it is by UV radiation, here they appear in different colors, so based on the similarity and binding hybridizations. So generally labeling here, as I mentioned, radioactive isotopes are most used. Other than that, very commonly used are fluorescence labeling, where combination of Psi3 and Psi5 are used. Psi3 is a green color laser is going to come out, and Psi5 red color is going to be obtained. So these are the steps. It includes first sample. They are going to purify the sample, means isolate the mRNA molecules. Then they produces the cDNA out of it, and then there will be coupling of means labeling is going to be done with the fluorescent label, and then the hybridization is going to be allowed to happen with the chip. And after hybridization there will be washing and scanning is going to be done, and finally whatever the data which is going to be obtained after the scanning, that will be analyzed for expression studies or presence of the this one. So you can see this is one microarray chip. So how it appears once the hybridization of is done with the target DNAs. So genome chip is nothing but the nucleic acid microarrays which are generated by using nucleotide probes. So these nucleotide probes are going to be allowed to get, means they are going to be attached to the matrix, which is usually a glass microscopic slide, or it can be a nylon, silicon, or nitrocellulose membrane also. Nowadays they are using Affymetric matrix also, and these oligonucleotides are going to be printed onto the matrix by using robotics or photolithography, or even people are using simply by using an inkjet printing technique also people are using in printing these particular or attaching the probes onto the chip. So you can see here the glass slide at the top, Affymetric matrix, which is actually as the gene chip. so which is being added by this robotic technology, and it appears like the other three. You can see that. So the types of array, depending upon the kind of immobilized samples, people can go for nucleic acid array or they can go for protein arrays also. In nucleic acid arrays, we can have cDNA array. Even people go for SNPs. They go for comparative genome hybridizations also. I'll be talking mainly here the nucleic acid hybridization or nucleic acid arrays, especially the cDNA microarray, which is used for studying the gene expression in a particular organism or a cell, can be done by this particular method. Means you can see here there is a normal cell, there is a cancer cell. You can take the samples, find the culture, made a cell culture, take an mRNA, total mRNA from all the means from the cells, and then that can be subjected for cDNA production. Those cDNAs can be labeled separately with either Cy5 or Cy3. So based on that, which will give the green and red fluorescence. Then those particular DNAs are going to be mixed and they'll be added onto the normal this chip. So once the hybridization is done, then you can subject for washing and you can subject for scanning. When we scan, it appears like this, which shows wherever there is gray color, that indicates there is no presence of those particular genes are not present in the cells. Wherever yellow is there, presence in both the cells, because yellow is going to appear when a combination of green and red is going to take place. Green indicates only it is in normal cell genes, and red indicates only the cancer cell gene expressions. So this is just an method where I am showing you can use it for studying the gene expression pattern in a normal cell and a cancer cell, and you can compare it. So this is how it's going to be. There's going to be a glass slide where you can label it. There will be array having a cover slip, lifter slips will be there. Then hybridization chamber will be there where and we can apply the sample at one end, which will slowly goes and spreads over all, and then hybridization is going to happen. Then later we will subject to washing and then scanning. So application wise, it is used in the study of gene expression profiling tool, where people are going to use this particular method to study the expression of genes, what are all genes get expressed in a particular organism and at different conditions, and how the gene expression varies with the physical and physiological conditions. And then this is also going to be used in comparative genomic tools also, where we can compare the expression of the genes between two different organisms, how it is similar genes expressed or different genes are going to be expressed in organisms. People are also using in diagnostics to identify the disease conditions. They are using it in drug discovery. How? Because the expression pattern or how exactly the organisms or the cell is going to behave in terms of gene expression during a in the presence and absence of drugs, and how these drugs are actually affecting the expression of a particular gene, which in turn can be used for developing a particular method of, or in turn can be used for evaluating at the basic, at the molecular level, the efficacy of a drug or efficiency of a drug. Then finally, toxicological research are also being done by using this microarray technology. So this is an application part of the recombinant DNA technology, wherein with the production of probes and by what I can say, by using the hybridization method, people are using for analysis. All the best, and if any doubts, please do contact. So this is about microarray technology. Thank you.